Hello everyone! Congratulations for reaching this far. I welcome you to Chapter 11, Learning, Memory, and Amnesia, How Your Brain Stores Information. So this chapter focuses on the roles played by various brain structures in the process of learning and memory. So this is a quick overview of what we're going to learn for this chapter. We are hoping that you already read Chapter 11. So let us begin! Amnesic effects of bilateral medial temporal lobectomy. Ironically, the person who contributed more than any other to our understanding of neuropsychology of memory was not a neuropsychologist. H.M. had the medial portions of his temporal lobes removed for the treatment of a severe case of epilepsy. I hope everybody has already read the case of H.M., the man who changed the study of memory. It's located on page 297 of this textbook. Now let's move on to the formal assessment of H.M.'s anterograde amnesia, discovery of unconscious memories. In this part, we will describe the specific memory tests used to assess H.M.'s anterograde amnesia. These are actually 5 tests that were used to assess his long-term memory. The first test was called the Digit Span +1 test. This is actually a classic test of verbal long-term memory. H.M. was asked to repeat 5 digits that were read to him at 1 second interval. Of course, he repeated the sequence correctly. On the next trial, the same 5 digits were presented in the same sequence with 1 new digit added to the end. After 25 trials, H.M. had not managed to repeat the 8-digit sequence. H.M.'s inability to form certain long-term memories was objectively illustrated by his performance on this test. The second test was called the Block Tapping Memory Span test. Take note that H.M. had global amnesia, that is an amnesia for information presented in all sensory modalities. So in this test, an array of 9 blocks was spread out on a board in front of H.M., and he was asked to watch the neuropsychologist touch a sequence of them and then repeat the same sequence of touches. H.M. had a block tapping span of 5 blocks, which was the normal range, but he could not learn to correctly touch a sequence of 6 blocks, even when the sequence was repeated 12 times. The third test was called the mirror drawing test. So H.M.'s task was to draw a line within the boundaries of a star-shaped target by watching his hand in a mirror. H.M. was asked to trace the star 10 times on each of 3 consecutive days. His performance improved over 3 days, which indicates retention of the task. However, despite his improved performance, he could not recall ever having completed the task before. This test was the first indication that his anterograde amnesia did not involve all long-term memories. The fourth test was actually called the incomplete picture test. This is a non-sensory motor test of memory that employs 5 sets of fragmented drawings. So each set contains drawings of the same 20 objects, but the sets differ in their degree of completeness. The subject is asked to identify the 20 objects from the most fragmented set. Then those objects that go unrecognized are presented in their set 2 versions, and so on until all 20 items have been identified. But despite of his improved performance, H.M. could not recall previously performing the task. Now the last test was called the Pavlovian conditioning. So our patient had learned an eye blink Pavlovian conditioning task. So a tone was sounded just before a puff of air was administered to his eye. These trials were repeated until the tone alone elicited an eye blink. 2 years later, he retained this conditioned response almost perfectly, although he had no conscious recollections of the training. Let us now proceed to the 3 major scientific contributions of H.M.'s case. First, by showing that the medial temporal lobes play an important role in memory, Henry's case spawned a massive research effort aimed at clarifying the mnemonic functions of the hippocampus and other medial temporal lobe structures. Second, it supported a theory that there are different modes of storage for short-term, long-term, and remote memory. Lastly, Henry's case was the first to reveal that amnesic patients might claim no recollections of a previous experience while demonstrating memory for it by improved performance. This discovery led to the creation of two distinct categories of long-term memories: the conscious long-term memories, which is known as explicit memories, and the long-term memories demonstrated by improved test performance without conscious awareness, which is known as implicit memories. People who has difficulty in forming explicit long-term memories while retaining the ability to form implicit long-term memories are symptoms of medial temporal lobe amnesia. Tests that assess implicit memory are called repetition priming tests. The incomplete picture test is an example of one. Now let us differentiate semantic and episodic memories. Explicit memories fall into two categories, and that many of these amnesics tend to have far greater difficulties with one category than the other. Explicit long-term memories come in two varieties. We have the semantic memories, which are explicit memories for general facts or information, and episodic memories, which are explicit memories for particular events, such as episodes of one's life. Global cerebral ischemia is someone who has experienced an interruption of blood supply to their entire brain. These people often suffer from medial temporal lobe amnesia. As we can see, there are actually four major components of the hippocampus. We have the CA1, CA2, CA3, CA4 subfields, and the dentate gyrus. Hippocampal damage by itself can produce medial temporal lobe amnesia. Arguably, the strongest evidence that selective hippocampal damage can cause medial temporal lobe amnesia comes from cases of transient global amnesia. In this case of transient global amnesia, the amnesia is transient, typically lasting only 4 to 6 hours. Let's move on to the amnesia of Korsakoff syndrome. So Korsakoff syndrome is a disorder of memory which is caused by thiamine deficiency and is commonly seen in chronic alcoholism. In its advanced stages, it is characterized by a variety of sensory and motor problems, extreme confusion, personality changes, and a risk of death from liver, gastrointestinal, or heart disorder. So the amnesia of Korsakoff syndrome is similar to medial temporal lobe amnesia in some respects. During the early stages of the disorder, anterograde amnesia for explicit episodic memories is the most prominent symptom. However, as the disorder progresses, retrograde amnesia also develops. But because the brain damage associated with Korsakoff syndrome is diffuse, it has not been easy to identify which part of it is specifically responsible for the amnesia. Amnesia of Alzheimer's disease. So Alzheimer's disease is another major cause of amnesia. The first sign of this disease is often a mild deterioration of memory. However, the disorder is progressive. Eventually, dementia develops and becomes so severe that the patient is incapable of even simple activities such as eating, speaking, or bladder control. So the brain damage associated with Alzheimer's disease is extremely diffuse, involving many areas including the medial temporal lobes and the prefrontal cortex, which play major roles in memory.
So let's move on to the next topic, which is the post-traumatic amnesia. So blows to the head that do not penetrate the skull but are severe enough to produce concussion are the most common causes of amnesia. So amnesia following a non-penetrating blow to the head is called post-traumatic amnesia. This time, let's summarize the effects of a close head injury on memory. So the coma following a severe blow to the head usually lasts a few seconds or minutes. But in severe cases, it can last weeks. Once the patient regains consciousness, he or she experiences a period of confusion. Testing usually reveals that the patient has permanent retrograde amnesia for the events that lead up to the blow and permanent anterograde amnesia for many of the events that occurred during the subsequent period of confusion. So this figure shows the summary of the effects of a closed head injury on memory. Gradients of retrograde amnesia and memory consolidation. Gradients of retrograde amnesia after concussion seem to provide evidence for memory consolidation. The classic theory of memory consolidation is Hebb's theory. He argued that memories of experiences are stored in a short term by neural activity, reverberating in closed circuits. Electroconvulsive shock seemed to provide a controlled method of studying memory consolidation. Electroconvulsive shock, or ECS, is an intense, brief, diffuse, seizure-inducing current that is administered to the brain through large electrodes attached to the scalp. So this time, let us differentiate consolidation and reconsolidation. So the case of Henry Molaison provided evidence of memory consolidation, and it seemed to suggest that the hippocampus played a special role in it. Scoville and Milner suggested that memories are temporarily stored in the hippocampus until they can be transferred to a more stable cortical storage system. This theory has become known as the standard consolidation theory or dual trace theory. Reconsolidation, on the other hand, is a theoretical construct that has recently attracted researchers' attention. The hypothesis is that each time a memory is retrieved from a long-term storage, it is temporarily held in LABA, short-term memory, where it is once again susceptible to post-traumatic amnesia until it is reconsolidated. Evolving perspective of the role of the hippocampus in memory. As interesting and informative as the study of patients with amnesia can be, it also has its major limitation. The first report of Henry Molaison's case in the 1950s triggered a massive effort to develop an animal model of his disorder so that it could be subjected to experimental analysis. In retrospect, there were two reasons for the initial difficulty in developing an animal model. First, it was not initially apparent that Henry Molaison's anterograde amnesia did not extend to all kinds of long-term memory. Second, it was incorrectly assumed that the amnesic effects of medial temporal lobe lesions were largely, if not entirely, attributable to hippocampal damage. So, animal models of the object recognition amnesia. There are actually two versions of the delayed non-matching to sample test. These are the monkey version and the rat version. But this time, we will first tackle the monkey version of the delayed non-matching to sample test. So in separate laboratories, Gaffan and Mishkin and Delacour showed that monkeys with bilateral medial temporal lobectomies have major problems performing long-term memories for objects encountered in the delayed non-matching to sample test. In this test, a monkey is presented with distinctive object, under which it finds food. Then after a delay, the monkey is presented with two test objects, the sample object and an unfamiliar object. The monkey must remember that the sample object so that it can select the unfamiliar object to obtain food concealed beneath it. So the correct performance of a trial is illustrated here. So this time, let us tackle the rat version of the delayed non-matching to sample test. So in order to understand why the development of the rat version of the delayed non-matching to sample test is important, let us examine figure 11.10. 11.10 shows aspiration lesions of the hippocampus in monkeys and rats. Because of differences in the size and location of the hippocampus, which is the pink one, in monkeys and in rats, hippocampectomy typically involves the removal of large amounts of medial temporal cortex, which is the red one, in monkeys but not in rats. So in monkeys, the hippocampus is usually removed by aspiration via the inferior surface of the brain, thus destroying substantial amounts of adjacent medial temporal cortex. On the other hand, which is in rats, aspiration of hippocampus is usually performed via the dorsal surface of the brain, thus destroying small amounts of parietal neocortex. So the version of the delayed non-matching to sample test for rats was developed by David Mumby using an apparatus that has become known as the Mumby box. Neuroanatomical basis of the object recognition deficits resulting from bilateral medial temporal lobectomy. Bilateral surgical removal of the medial temporal cortex consistently produces severe and permanent deficits in performance on the delayed non-matching to sample test and other tests of object recognition. In contrast, bilateral surgical removal of the hippocampus produces only modest deficits, and bilateral destruction of the amygdala produces none. This figure compares the effects of medial temporal cortex lesions and hippocampus amygdala lesions on object recognition memory in rats. So for this chapter, you have seen that our perspective of the role of the hippocampus in memory has changed since the first published report of HM's case. Initially, the hippocampus was thought to be the site of temporary storage for all newly formed memories. However, it was soon discovered that the structures of the medial temporal lobes have a more specific function. They appear to play a major role only in explicit episodic memories.
Neurons of the medial temporal lobes and memory. Now let's go to the hippocampal place cells and entorhinal grid cells. Place cells are neurons that respond only when the subject is in specific locations. One line of research on hippocampal place cells has focused on the entorhinal cortex. This is an area of the medial temporal cortex that is a major source of neural signals to the hippocampus. So how hippocampal place cells obtain their spatial information come from the discovery of so-called grid cells. These are entorhinal neurons that each have an extensive array of evenly spaced place fields producing a pattern reminiscent of graph paper. Two lines of evidence suggest that the response of hippocampal place cells depend on the input from entorhinal grid cells. First, there is a major pathway from entorhinal cortex to the hippocampus, and second, entorhinal grid cells respond relatively reflexively to location, whereas hippocampal place cells respond to place in combination with other features of the test environment. The comparative studies of hippocampus and spatial memory. There are evidences that the hippocampus plays a role in spatial memory. Studies show that food-catching birds tend to have larger hippocampuses than related to non-food catching species. Recently, the hippocampus has been implicated in several forms of memory other than spatial memory. For example, certain cells in the hippocampus have recently been shown to code for the temporal aspects of experience, which are called time cells. The hippocampus has been shown to play a role in learning about social organization, or known as social space. Jennifer Aniston neurons, concept cells. As you have previously learned, the major structures of the medial temporal lobes are the hippocampus, amygdala, and the medial temporal cortex. The medial temporal cortex is composed of the entorhinal, perirhinal, and the parahippocampal cortices. In one of the first neurons to be studied in this way, the neuron fired in response to images of the actress Jennifer Aniston, but not to 80 other images, thus the name Jennifer Aniston neurons. These neurons are highly selective. Each neuron responds to only a small number of test objects or individuals. The most remarkable feature of this neuron is that they respond to ideas or concepts rather than to particulars, which is why they are also called as concept cells. Now let's move on to engram cells. If you recall, in optogenetics, neuroscientists insert an opsin gene into particular neurons, after which they can then use light to either hyperpolarize or depolarize those neurons. The identification of an engram cell via optogenetics is typically a two-stage process. In the first stage, the tagging stage, the neurons that are activated during the learning task are induced to express opsins while an animal engaged in a particular learning task. In the second stage, the manipulate stage, the previously active neurons are now either inhibited or excited by using light to influence the activity of the opsin tag neurons. Where do you think are memories stored? So far, this chapter has focused on four neural structures that appear to play some role in the storage of memories. The hippocampus and the medial temporal cortex have roles in episodic memory, and the mediodorsal nucleus of the thalamus and the basal forebrain have been implicated in the memory deficits of Korsakoff's and Alzheimer's disease, respectively. Now, we take a look at 5 other areas of the brain that have been implicated in memory. The 5 brain areas implicated in memory. Infratemporal cortex. So areas of the brain that are active during retention of an experience tend to be the same ones activated during the original experience. In particular, attention has focused into infratemporal cortex, which is the cortex of the inferior temporal cortex. Researchers have argued that the inferior temporal cortex, in concert with adjacent perirhinal cortex, plays an important role in storing memories of visual input. Next is the amygdala. The amygdala is thought to play a special role in memory for the emotional significance of experience. The amygdala might be the reason why emotion-provoking events are remembered better than neutral events. Next is the prefrontal cortex. This is the area of the frontal cortex anterior to motor cortex. So different parts of the prefrontal cortex play different roles in memory. Some regions of the prefrontal cortex seems to perform fundamental cognitive processes during working memory tasks, and other regions of prefrontal cortex participate in other memory process. Lastly, the cerebellum and the striatum. The cerebellum is thought to participate in storage of memories of learned sensory motor skills through its various neuroplastic mechanisms, whereas the striatum is thought to store memories for consistent relationships between stimuli and responses, the type of memories that develop incrementally over many trials. Sometimes this striatum-based form of learning is referred to as habit formation.
So this time, let us discuss long-term potentiation. In 1973, Bliss and Lumu showed that there is a facilitation of synaptic transmission following high-frequency electrical stimulation applied to presynaptic neurons. This phenomenon has been termed as long-term potentiation, or LTP. LTP has been demonstrated in many species and in many parts of their brain, but it has been most frequently studied in the rodent hippocampus. So this figure shows a slice of rat hippocampal tissue that illustrates the three synapses at which LTP is most commonly studied. Number 1 is the dentate cranial cell synapse. Number 2 is the CA3 pyramidal cell synapse. And number 3, the CA1 pyramidal cell synapse. LTP is among the most widely studied neuroscientific phenomena. The question is, why? The reason goes back to 1949 and Hubb's influential theory of memory. The synaptic changes that Hubb hypothesized as underlying long-term memory seem to be the same kind of changes that underlie LTP. So LTP has two key properties that Hubb proposed as characteristics of the physiological mechanism of learning and memory. First, LTP can last for a long time, for several months after multiple high-frequency stimulation. Second, LTP develops only if the firing of the presynaptic neuron is followed by the firing of the postsynaptic neuron. Conceiving of LTP as a three-part process, many researchers are investigating the mechanism of induction, maintenance, and expression. That is, the processes by which high-frequency stimulation induce LTP, the learning, the changes responsible for the maintenance of LTP, which is the memory, and the changes that allow it to be expressed during the test, which is the recall. So this time, let us discuss the induction of LTP learning. The NMDA, or N-methyl aspartate receptor, is prominent at the synapses at which LTP is commonly studied. The NMDA receptor is a receptor for glutamate, the main excitatory neurotransmitter of the brain. So an NMDA receptor does not respond maximally unless two events occur simultaneously. First, glutamate must bind to it, and the second, postsynaptic neuron must already be partially depolarized. So this dual requirement stems from the fact that the calcium channels associated with NMDA receptors allow only small numbers of calcium ions. So the figure shows the induction of NMDA receptor-mediated LTP. So let us explain. If one glutaminergic neuron were to fire by itself and release its glutamate neurotransmitter across a synapse onto the NMDA receptors of a postsynaptic neuron, there would be no potentiation of transmission at that synapse because the postsynaptic cell would not fire. However, if the postsynaptic neuron were partially depolarized by input from another neurons, when the presynaptic neuron fired, the binding of the glutamate to the NMDA receptors would open wide. Calcium ions would flow into the postsynaptic neuron, and transmission across the synapses between the presynaptic and the postsynaptic neurons would be potentiated. So, the search for the mechanism underlying the maintenance and expression of LTP began with attempts to determine whether these mechanisms occur in presynaptic or postsynaptic neurons. This question has been answered. The maintenance and expression of LTP involve changes in both presynaptic and postsynaptic neurons. LTP maintenance and expression has contributed to several important discoveries. Here are the three of them. First discovery is that it was clear that there must be a mechanism for keeping the events at one set of synapses on a postsynaptic neuron, thus affecting other synapses on the same neuron. This specificity is due to dendritic spines. Second discovery is that it became apparent that maintenance of LTP involves structural changes, which depend on protein synthesis. The discovery that LTP causes structural changes was exciting because the structure of neurons and neural circuits had been assumed to be static. Third is the discovery of structural changes in neurons following the induction of LTP stimulated a search for a mechanism by which a neuron's activity could change its structure. This led to the discovery of numerous transcription factors. So now let's move on to the next topic, which is the variability of LTP. So most of the research on LTP has focused on NMDA receptor-mediated LTP in the hippocampus. It is now clear that NMDA receptor-mediated LTP involves a complex array of changes that are difficult to sort of. There is also LTD, which is the long-term depression, which is the flip side of LTP and occurs in response to prolonged low-frequency stimulation of presynaptic neurons. And then there is metaplasticity, which refers to the fact that LTP and LTD induction can be modulated by prior synaptic activity. So, a full understanding of LTP and its role in memory will require an understanding of LTD and metaplasticity. So, have you heard of a word infantile amnesia? Or do you know that we all experience this kind of amnesia? So, infantile amnesia, that is, we remember virtually nothing of the events of our infancy. So, there are some studies conducted in regards to infantile amnesia. So, one study is that children were shown a series of photographs of preschool-aged children, some of whom had been their preschool classmates. So, whether they explicitly remember a former classmate or not, they consistently displayed a large skin conductance response to the photographs of their former classmates, but not to the control photographs. So, in a second study of infantile amnesia, Drami and Yukumbi used a version of the incomplete pictures test. So, there are actually 3 groups, and all these groups displayed substantial implicit memory. All participants were able to identify the drawings they had previously seen sooner, even when they had no conscious recollection of them all. So this time we will discuss the findings on the efficacy of smart drugs. Nootropics, or what you call the smart drugs, these are substances that are thought to improve memory. Perhaps you have heard of or even tried some of them. These are the ginkgo biloba extracts, ginseng extracts, multivitamins, glucose, and many more. So the evidence that nootropics enhance memory has been reviewed several times by independent scientists. However, there is no supported evidence that nootropic has shown to have memory enhancing effects. There may be enough positive evidence to warrant continued investigation of some potential nootropics, but there is not nearly enough to justify the various claims that are made in the advertisement for some substances.
So, before we end our discussion, we will have a little bit of a review so that we'll be able to remember the things we have discussed. So, let's scan our brains, shall we? Any pathological loss to memory is called, if you answered amnesia, then you're correct. Blank is a disorder of memory common in people who have consumed large amounts of alcohol. If your answer is Korsakoff syndrome, then you are correct. Last question. Identification of an engram cell is a two-stage process involving the blank and manipulate stages. If you answered tagging, then you are correct. Thanks for watching!